everyone, and welcome to today's lecture. What you will notice about these lecture videos is that for each slide that I cover, I'll first narrate the important information explaining the topic on that slide, and then I'll turn to the next slide, which will simply say Chalkboard Notes, and it will be a copy of what I would have written on the chalkboard had we been in the classroom. That way, for each of the slides, you don't have to overwhelm yourself trying to copy down every word that I'm saying. Instead, the most important notes that I would have written on the chalkboard will be written on the next slide. Today's lecture is going to focus on the citrate cycle. This is a very important cycle because it's basically the hub of cellular metabolism. This means that it's central to aerobic metabolism and to ATP production. And so you'll notice throughout this course that a lot of the other topics that we talk about in later lectures will kind of connect back to the citrate cycle. And you can see that a bit on this slide. Now, when we talk about the citrate cycle, we won't always call it the citrate cycle. Sometimes it will be called the Krebs cycle or TCA for the tricarboxylic acid cycle. Okay, so if you hear the name the Krebs cycle or TCA cycle, that's the same as saying the citrate cycle. Now, as I just mentioned a moment ago, this cycle, as you can see in this figure, connects to a lot of different processes that we're going to talk about throughout this course in later lectures. Now, one of the terms that I want you to know and circle star highlight is amphibolic, okay? Because like I just mentioned, this cycle connects to various processes, and this includes catabolic and anabolic processes or pathways. And what that means is catabolic. For that term, I want you to picture a cat scratching the crap out of a sofa. Okay, their little claws coming out and they're scratching, tearing that apart, breaking down that sofa with their claws. Okay, if you picture that every time you see or hear the word catabolic, it will remind you that catabolic pathways are degradation pathways, pathways in which you're breaking something down, such as breaking down a protein into its amino acids. Okay, now the opposite of that is anabolic. Anabolic, and I always underline the A in that and make people think Arnold Schwarzenegger for that one, because anabolic is like when you hear about anabolic steroids, that you're all about those gains, and anabolic processes are ones that will build something up. Okay, so the catabolic process I mentioned a moment ago is breaking down a protein into amino acids. The anabolic process would be building, connecting amino acids to make a protein. Okay, and you'll notice that in this figure here, the citrate cycle is connected to both types of pathways. Okay, you see that the arrows go in both directions. So we call it amphibolic because it's involved in both catabolic and anabolic pathways, breaking things down and also building them up. As I mentioned before, each slide will then have one of these chalkboard notes. Normally, I won't narrate on top of it, but I just wanted to let you know that when you see these appear, you can pause the video and jot down these notes to then study at a later time. Now, when we talk about the citrate cycle or any cycle, it's important to think about where exactly is it occurring. And the citrate cycle is going to occur in the mitochondrial matrix. Okay, and every meme has told you that the mitochondria is the powerhouse of the cell. Now, when you look at the citrate cycle, we say that the primary function is to oxidize acetyl-CoA. So let me just put the little laser on so that you can circle that term there, acetyl-CoA. The purpose of this cycle will be to oxidize that. And during this process, each turn of this citrate cycle is going to end up transferring 8 electrons to produce these various products that you see here, especially noting the fact that you're producing NADH, FADH2, and GTP. And over here in the figure, you can see the 8 electrons that I just mentioned a moment ago. Now, what's important about these products that you see here is when you look at the citrate cycle, those products that it makes, such as NADH and FADH2, they then feed into the electron transport chain and the ATP synthase complex, which means that from these products, you can then make a ton of energy of ATP. And we're going to go over that in later lectures, but I kind of like to point out that when you are looking at a cycle like the citrate cycle and you don't really see, a lot of ATP being made in that cycle, you may say, well, why? What's the point, if it's not going to give much energy? Well, the point is that it gives you products such as NADH and FADH2 that are money in the bank. They go toward the next steps of aerobic respiration, and you could end up making a lot of ATP in those next steps. Now, as I was just mentioning, that concept of things like money in the bank, some of the things that you will keep hearing in these lectures and in biochemistry are NAD plus, FAD, and then their alternate forms NADH and FADH. Now, the purpose of these is that they are very important in each of the different reactions that we're going to talk about throughout biochemistry. Now, they are what's called coenzymes. And first, I want you to circle star highlight NADH and FADH. These two, their purpose will be to donate electrons. Okay, donate electrons for ultimately making ATP. Whereas these two are the opposite, okay? These two, NAD plus and FAD, these will accept electrons in some of the reactions that we go over, okay? Now, the way that you can remember that is when you see NADH or FADH, you notice that they have this extra H at the end. So you can use that extra H to remind you that when you get rid of that, you are now donating electrons. Whereas these guys here, they don't have that extra H, so they can accept electrons. Okay? Now, this will be occurring in what's called oxidation reduction reactions. So those are the redox reactions that you've already seen in some of the previous lectures of biochemistry, but you'll keep seeing them throughout all of the other lectures. And redox reactions, you always think of that Leo says grr kind of trick to remembering, losing electrons is oxidation, gaining electrons is reduction. Now, another point that's very important on this slide is the question, where are they derived from? So NAD and FAD, or NADH and FADH, they're derived from vitamin B. Okay, so NAD is derived from niacin, which is vitamin B3, and FAD is derived from riboflavin, which is vitamin B2. Okay, and they're going to be very important in various reactions that we talk about in later lectures. Now, before the citric acid cycle, something else has to happen first. Now, we mentioned that the citrate cycle is going to be responsible for oxidizing acetyl-CoA. Well, that means that first you have to make that acetyl-CoA, right? So the enzyme that's necessary before the citric acid cycle is going to be this one here, pyruvate dehydrogenase. It is actually a complex of 3 different enzymes that you see here. Okay, it is a complex of the pyruvate dehydrogenase component, also a dihydrolipoyl transacetylase component, and a dihydrolipoyl dehydrogenase component. But you're not going to have to memorize any of those terms. I just want you to know the main term of the complex, which is pyruvate dehydrogenase. So circle star highlight that enzyme. And the function of that enzyme is to convert pyruvate to acetyl-CoA. And then that acetyl-CoA will go into the citric acid cycle. Now, I also want you to circle the delta G on this slide, that negative 33.4, because that will come back in a later slide when I ask you about the energetics of this process. Okay, so when you see the negative delta G, that tells you it's a highly favorable reaction. It is exergonic, and we're going to talk about that a little later. Now, with any process in biology or in life, you have to be able to control that process. You need to be able to control when it's occurring, so be able to activate it or deactivate it or repress it. And so a lot of times the way that we do that is based on the function of the enzyme. So now we just mentioned the enzyme pyruvate dehydrogenase. Okay, and that's going to again be converting pyruvate to produce acetyl-CoA. Now, this enzyme requires 5 coenzymes listed here in order for it to be active and functioning. And a lot of times what you will notice in biology is whenever you have an enzyme controlling a reaction, one of the best ways to inhibit or control that enzyme is through negative feedback. So you look at the process that that enzyme is controlling, and you look at the products and think about it. If you already have a lot of the products present, then you don't need that enzyme. Why am I going to waste time, energy, resources, you know, to do this reaction and get more products when I already have them? So the products a lot of time will be what inhibit the actions of that enzyme. So pyruvate dehydrogenase, for instance, is inhibited by acetyl-CoA, NADH, and also you'll notice that in this reaction occurring, we mentioned that one of the coenzymes is called TPP. And this figure over here shows that TPP is an enzyme that leads to lipoamides, okay? And so if you inhibit those as well, you are then inhibiting this process of, pyruvate dehydrogenase activity. And so one of the other inhibitors of PD that I want you to know, or inhibitors of pyruvate dehydrogenase, is arsenic, okay? Arsenic is something that irreversibly blocks the catalytic activity of lipoamide containing enzymes with covalent bonds, and so you cannot get the synthesis or the production of lipoamides. And so arsenic will block this pathway as well. And the other inhibitor that I want you to know is ATP, because again, the whole point of aerobic respiration and things like the citric acid cycle is to ultimately make ATP. And so if a cell has plenty of ATP, you're not going to want to waste resources trying to make more. Now, my favorite parts of biochemistry are the medical connections, especially ones that have to deal with food because I love food. Now, we just mentioned a moment ago that pyruvate dehydrogenase, one of the coenzymes that's needed for pyruvate dehydrogenase, or we'll call that enzyme PD, to work is TPP. And what TPP stands for is thymine pyrophosphate. Okay, now in order to get that coenzyme that allows pyruvate dehydrogenase to work, what the cell needs to do is dietary thymine has to be phosphorylated by an enzyme and ultimately generates the active coenzyme TPP. And you then need that TPP for pyruvate dehydrogenase to then convert pyruvate to acetyl-CoA, which then will be able to feed into the citrate cycle. Now that brings us to the medical connection of beriberi. Beriberi is a disease that's characterized by things like anorexia, which means you lose your appetite, cardiovascular issues like tachycardia. And what tachycardia means is that racing heartbeat that you can get sometimes. And also neurological problems. So people will end up, if they have beriberi, they'll have difficulty walking, or they could get paralysis or even numbness. And what it's actually caused by is a thiamine deficiency. And thiamine is vitamin B1. Okay. Now, when you think about this disease, there are a few things that lead to it. Okay. And I have the figure on the side. Let me just pull up this laser. The figure on the side shows, for instance, white rice that's been polished can lead to beriberi because the milling of that rice, the milling from the whole dark grain to make the polished white rice, that actually removes bran, and bran is what contains the thiamine in that rice. So if they're eating a whole bunch of polished white rice, they're not getting enough thiamine, which means you end up deficient, which gives you beriberi. Another thing that can cause beriberi is places that have diets that are high in raw fish or silkworms. And I know you look at a silkworm and you're like, ew, that's disgusting. But it's actually a delicacy in a lot of places, and it's a great source of various nutrients. Now, these two things here, raw fish and silkworms, they have high levels of enzymes called thymidase. Okay, and if you see an enzyme like thymidase, that tells you that it's going to be breaking down thymine. Okay, and if it's breaking down thymine, you're then deficient in thymine and you get beriberi. Now, the reason why I emphasize the fact of raw fish, or even saying, raw silkworms, what happens is if you instead cook them and eat them cooked, you're now destroying the enzymes, because heat, as you know, denatures proteins, and enzymes are proteins. So that means that cooking things like fish will protect the thymine because you have denatured the enzymes that would break it down. Okay, so please remember beriberi is ultimately thiamine deficiency, and things like white rice or raw fish and silkworms are able to lead to it for the reasons that I just mentioned. Now, this slide gives you a summary of the reactions that will occur in the citrate cycle. The overall pathway is called an aerobic pathway. So that's the type of pathway is aerobic, meaning that it requires oxygen. Keep in mind, glycolysis does not require oxygen, but then the citrate cycle, the electron transport chain, and ATP synthase complex all are aerobic pathways requiring oxygen. And then you will notice how many reactions. There are 8 reactions in total to the citrate cycle. The first of the reactions in this cycle is going to be the citrate synthase reaction. Okay, so for each of these slides that we go over that are the reactions, you'll get to see that we call the reaction based on the enzyme that's responsible for this reaction. So the first one is citrate synthase. I want you to circle star highlight the name of that enzyme because it's an important regulator of the overall process of the citrate cycle. Now you'll notice that in this step, citrate synthase is responsible for, well, what do you think? The synthesis of citrate. Now, if you already have a lot of citrate, then the cell is able to bypass this first reaction and go straight into the next reaction that we're going to talk about. Okay, so for instance, if you are someone that drinks a ton of orange juice every day, well, then you would be able to bypass step one because you already have the citrate that then feeds into the next step. Okay, and so you would be able to have elevated citrate feeding into future steps of the process. Now, the type of reaction that is occurring here is a highly favorable condensation reaction. And when we say highly favorable, it's because the delta G in this process would be negative 31.4 kilojoules per moles. And so the negative is of the delta G is a great benefit when you have a highly favorable reaction because it encourages the process or that reaction to occur. The next reaction is the econotase reaction. So again, you'll see the enzymes listed on the figures. So it's the econotase reaction. And the type of reaction occurring here is called an isomerization. And what that means is you'll see a conversion or a shuffling of the organization of the atoms in the molecule, but you are not losing or gaining any of the number of those particular atoms. So you see how things get moved around. So this hydroxyl group gets shifted down to here, but you're not actually changing the number of carbons, of hydrogens, or of oxygens in that reaction. And this enzyme is an iron sulfur cluster enzyme. And what you see it doing is a two-step process with an intermediate here. And the two-step process is dehydration, hydration. Okay, so dehydration being the loss of a water molecule, and then hydration being the gain of a water molecule. Now, I know you look at that and you're like, okay, that sounds like nothing is getting done. Okay, nothing getting done, which is basically what my undergrads do. But anyway, so dehydration, hydration sounds like you're not doing anything, right? But in actuality, note the fact that you don't want to gain or lose any atoms. You just want to shift the location of where those atoms are, okay, the organization of them. And so that's why it's important to have this dehydration, hydration occurring here. The next reaction, reaction number 3, is the isocitrate dehydrogenase reaction. This is the type of reaction called oxidative decarboxylation. And decarboxylation is seen right here. It's the removal of carbon dioxide. This is going to be the first carbon dioxide lost in the citrate cycle. And what's also important is that you'll notice that NADH or NADPH is produced in this step. And NADH getting produced is always an important thing because, like I said earlier, that is money in the bank that can then go toward the electron transport chain and ATP synthase later on to create more energy. Reaction 4, this one is the alpha ketoglutarate dehydrogenase complex. Okay, so you see it's a complex of 3 enzymes, meaning it's a multi-subunit enzyme. And this is another example of the type of reaction called oxidative decarboxylation. So again, decarboxylation because you see that carbon dioxide is released here. And also what's important is, again, you are producing more NADH. And once again, NADH, that is money in the bank for later on reactions. The fifth reaction is called the succinyl-CoA synthetase reaction, which you see over here. This is substrate level phosphorylation, and when you hear substrate level phosphorylation, a lot of times, even with the same enzyme, you see ATP produced, but in this case, you are generating GTP instead, but it's still a form of energy and useful in later on reactions that we're going to see in future lectures. So again, another idea of money in the bank, which I'm just going to keep saying here. The next reaction that you see here is succinate dehydrogenase. Okay, so that's over here. There are a few things I want to point out. First of all, I want to mention that the type of reaction is a redox reaction, which I mentioned earlier. Redox means reduction oxidation. So again, the idea that one thing is losing electrons, the other is gaining electrons. And what's important here is, again, that idea of money in the bank. You see the production of FADH2. And FADH2 is going to be important for going into later processes, later pathways, such as the electron transport chain. And then ATP synthase complex comes next, and you're able to produce quite a bit of ATP when you go down those pathways. The next reaction, which you see here, is fumarase. In the previous reaction, you might have noticed on the figure that fumarate was produced, and so now fumarase is going to lead to the conversion of fumarate into malate, and you're going to see that term come up in later lectures as well, as being important for some of the other cycles or pathways that we talk about. Now, this type of reaction is called hydration, and you'll notice that you have hydroxyl and hydrogen ion, and so you end up with H2O, so water hydration going into this process to ultimately lead to malate. The last reaction that we're going to mention is malate dehydrogenase. This is an oxidation type of reaction. And a couple things I want to point out. So first of all, I want you to note again, NADH is getting produced here. And anytime you see NADH or FADH2, please remember that concept of money in the bank. That will be important. Electron donors, coenzymes in later reactions, later processes that then allow for the production of ATP. So in each of these reactions where you see NADH and FADH2 being produced, you don't see ATP as the product here, but ultimately those coenzymes will lead to the production of ATP in later steps. Now, the other thing I want to mention here is the terminology that you see on this slide. So looking at malate and oxaloacetate, I want you to remember those two names because they're going to be important in later on lectures looking at different pathways and the movement of intermediates throughout the cell, because certain forms of intermediates can't pass through certain membranes. And so you're going to see that sometimes you need conversion, let's say to malate or to oxaloacetate, depending on where you are in the cell and where you have to go. Okay, so just keep in mind that you will see those two terms come up again in later lectures. So now earlier I mentioned that we would talk about the energetics of the citric acid cycle. And so we look into whether something's exergonic or endergonic. Again, these are terms that you saw in previous lectures, but we'll also see coming back in later lectures as well. Just a reminder, when a delta G is negative, such as the first one here, that means it's exergonic and highly favorable. So that tells you that the cell will want to do that reaction, okay? If instead you see endergonic reactions, those will be the ones that have a positive delta G. And even though it's not highly favorable, you can still get those reactions to occur by coupling or pairing it with exergonic reactions. And so what you'll notice in the process here, we do have some positive values, some endergonic reactions, but they still end up occurring because they are paired to these other reactions that are highly favorable exergonic reactions. And the overall process is highly negative, which means all of these steps will want to occur, okay? Because you have paired together the unfavorable with the favorable. Now, to answer the two questions that we have here, first, we have the conversion of pyruvate to acetyl-CoA. Again, please remember that this is the reaction that has to occur before the citric acid cycle, and it is done by the pyruvate dehydrogenase, the PD enzyme that we talked about earlier. You'll notice that the delta G is negative 33.4, which tells you that it is exergonic and highly favorable. We then look at the citric acid cycle, which are the 8 steps here. If you took the delta G, the free energy values for those 8 steps, and added them together, the negatives and the positives, you would end up with negative 44.3, and that would mean that it is also exergonic, highly favorable. And so the overall process as well of that pre-citric acid cycle step plus the 8 steps of the citric acid cycle, you see over here, negative 77.7, it is all exergonic, highly favorable pathways. Now, like I mentioned earlier, when we talk about any process in biology or anything in life, you need to be able to control it. It has to be highly regulated. You're not going to want to, a whole bunch of products being made during times when there's already a lot of those products. It would be wasteful, and the way I over exaggerate it because I am dramatic is just picture the poor little cell bursting and exploding because there's just too much product. So when you look at things occurring like the citric acid cycle, you'll notice that a lot of times the inhibitors of the process are the products. So having a lot of certain products will prevent certain steps from occurring. And the opposite of that, activators, if you have a lot of the reactants, let's say you have a lot of pyruvate, then you are going to activate the pathway because it will want to then break down that pyruvate and go through the whole cycle. Okay, so when we look at the logical inhibitors and activators, the reasoning behind it is for what I just said. If they're a product, it will be inhibitory. You don't want to be wasteful. If it is a reactant, then you're going to have it activating the pathway because, we have it, let's use it. Okay. Now, when you look at this process, you'll notice certain things such as the citrate that we mentioned before. If there's a lot of citrate present, such as drinking all that orange juice that I mentioned earlier, getting a lot of citric acid, well, then that will inhibit the first step of the process. But because it's a cycle, okay, that citrate is actually also a reactant. It's not just the product, it's the product of the first step, but it's the reactant in the second step. So you'll end up having citrate inhibit the first step, but it will activate the second step. So that citrate that you have from your diet will then feed into the later steps of the cycle. Okay. Other inhibitors would be things like ATP and NADH, because we said in this process you saw multiple steps wanted to produce ATP or NADH, and when you have that, you have those products, you don't need to activate the cycle. It would be wasteful to have these steps occurring. Okay, now I want you to notice over here, we have cells in resting metabolic state versus cells in active metabolic state. So if the cell is in a resting state, you notice it's, think about it as you're resting. You don't need energy if you're resting, right? So you'll see a high ATP, high NADH levels. And what do we know about those? Well, if we have a ton of ATP and NADH, why would we have to do the citric acid cycle? We already have them. It would be a waste of time and energy. Okay, so in these cases, in a resting metabolic state, the citric acid cycle will be inhibited. Whereas the opposite is the highly active metabolic state. In this highly active state, you need more energy. Think about it. If you're active, you're going to the gym, you're working out, you need more energy to do that, right? So it's the same thing with the cell. It needs more energy. The way that you'll see this is that it's low in ATP and it's low in NADH, which means you want to make those things, which means you will want the citric acid cycle activated. Okay, so these here will activate the cycle. Okay, so now this is one of the terms that I told you to circle, star, highlight, which is amphibolic. Make sure you remember the definition means that this is a pathway that has both catabolic and anabolic parts to it, and it's involved in other anabolic and catabolic pathways. So now, when we ask why exactly is this particular cycle considered amphibolic, well, if you look at this figure, you can see a lot of the different connections that the citric acid cycle has to other pathways and other processes, including catabolic and anabolic ones. So, first of all, to remind you what those terms mean, catabolic means that something's being broken down. And so, for instance, in this pathway, if you're breaking down acetyl-CoA to obtain carbon dioxide and water and release a large amount of energy, well, then that is a catabolic part of this pathway. But then at the same time, you notice that a lot of the intermediates in this pathway then go toward various biosynthesis pathways. Okay, so you can see that, for instance, uh, the intermediates can provide or be precursors for biosynthesis of things like amino acids or for glucose, okay, for fatty acids and cholesterol. So you have both processes being part of this cycle. Another term that I want you to know is anaplerotic reactions. And so you can tell if I like a word because I think it sounds cool, odds are you will get asked about it or reminded of it again at another time. So, if you want to sound really cool with your friends, just drop the word anaplerotic. Okay. It does have the erotic in it, so I'm sure your friends will listen. When you think of this word, the meaning is that these are reactions that can replenish any metabolic intermediates. Okay, so if a reaction is replenishing intermediates of the citrate cycle, such as oxaloacetate or malate, then those reactions are considered anaplerotic because those are intermediates of the citric acid cycle. Okay, so for example, on this slide, pyruvate carboxylase is an enzyme for an anaplerotic reaction because it leads to the synthesis of oxaloacetate, which is an intermediate in various different reactions or processes. Okay, either of these guys here, phos- phosphoenol pyruvate carboxylase or malic enzyme, both of these, which are replenishing oxaloacetate and malate, which we saw are both part of the citrate cycle. These reactions here would be considered anaplerotic, okay? So again, it's any reaction that is replenishing metabolic intermediates. That now brings us to the end of this lesson, and I have two review questions for you to practice here. Instead of giving you the answers, I would like you to, based on this lesson, try to answer them yourself, and then please send me the answer in the Remind app. If you've forgotten what the code is, please check it on the syllabus or send me an email, and I'll be able to get that for you. Just a reminder, whenever I give you review questions like that and ask you to send them to me in the Remind app, please don't cheat. Don't just ask your friends for the answer because that is basically useless. You're not getting graded on it. I honestly don't care if you get it right or wrong at this moment. What I do care is that if you don't understand the topic and you are getting that question wrong, I want to be able to find that out now and help you and re-explain it in a different way to, one-on-one in the Remind app, kind of correct it now so that you don't end up making the same mistake on an exam or on the final or in the future. Let's say you're taking standardized tests such as the MCAT. Okay, so you're only hurting yourself if you just ask a friend and send me whatever they say. Okay? And that is it for today. As always, if you have any questions, please send me a question in the Remind app through email, anytime, anything you need. I always tell people I have no life, so contact me anytime. If you are watching this and studying at 3 in the morning and you're afraid, like, oh no, I don't want to wake her up, don't worry about it. Please send the message while you are watching this. It's not going to wake me up, it's not going to disturb me. But if you wait, and figure, oh, I don't want to bother her right now. If you wait, you're probably going to be like me and completely forget to ever ask it. Okay? So never hesitate to ask me. That's it, and have a great day.